Hello viewers, my name is Damilare. In this lesson, we'll be looking at the topic supporting tissues in plants. Supporting tissues in plants. Plants need supporting tissues to remain upright. When you see a tree standing, it is supporting tissue that is holding it. When you see their branches spread out, it is the supporting tissue holding them. The leaves that are attached to the branches is the work of supporting tissues. And why? So that they can trap sunlight. The main supporting tissues in plants, they are the parenchyma, collenchyma, sclerenchyma. You can just use PSC like Pascal to remember P, parenchyma, S, sclerenchyma, C, collenchyma, so Pascal. Sclerenchyma is divided into fiber and sclerid. You can see selfish, SF and S, SFS. That's selfish. S sclerenchyma, F fiber, S sclerid. The xylem and phloem, they are also supporting tissues. Now let's focus on parenchyma. Once you hear parenchyma, what should first come to your mind is the word parents. Parents. You know, parents, they go extra mile. They are everywhere for their children. So meaning that parenchyma, they are the most common, most abundant tissues. We can find them in organs of higher plants. Meaning that parents, we see parents as higher higher humans. So it means parenchyma, you see them in higher plants. Parenchyma again, their cells occur in cortex, in pith, stem, roots, leaf, mesophyll, succulent storage tissues, and vascular tissues. That means xylem and phloem. How will you remember this? When you draw a tree, right from the top we have leaf. It means in the leaf of a tree you have parenchyma. If you are coming down through the branch, you will see the leaves. So the leaves, you see parenchyma there. Then when you come down here, let's see this place as a place where there is fruits, and fruits as if they are storing food there. So meaning that in the storage organ, you also see the parenchyma. Then when you keep coming down, see the center of this tree as pith. In the pith you see parenchyma. Then see this curve as cortex. In the cortex you see parenchyma. Then this one that looks like V, see it as vascular bundle. So meaning that in vascular bundle you also see parenchyma. Then when you come down, see these roots as also a place where you can see parenchyma. Now another thing is the parenchyma generally, if you want to summarize their functions, one, it stores food and water. Remember that our parents, they always try to get food and water for us. So meaning that parenchyma, they store food and water. Then their cells contains chloroplasts. You can use letter P of the parenchyma to remember photosynthesis. It means they can undergo photosynthesis because of the chloroplast they have. Then another thing is letter T, turgidity. From parenchyma, you can remember parents as T. So use the T to remember turgidity, meaning that they provide and maintain turgidity. Why? You know, turgid, when something is soaked with water. So since parenchyma, from the word parents, they try to get water for their children, it means they also, parenchyma, will make sure the plant is soaked with water. That is turgidity of the stem. Another one, it allows air to diffuse. Now from the spelling of parenchyma, you can still get some things to remember. From parenchyma, letter P to remember photosynthesis. The AR, if I put I there to turn to hair, means that for air there is diffusion. It means parenchyma also, it allows air to diffuse among cells. Then apart from that, letter C, you can use it to remember color. So that means absence of color. Then if you pick the letter MA at the end, manufacture, it means it can manufacture food. So, and finally, when parenchyma cells are less turgid, that means once there is no enough water, they have to drop leaves so that they can conserve water. It means they also helps to conserve water. So you can use the letter C of the parenchyma, that means they conserve water. Now let's move to collenchyma. If we use parents to remember parenchyma, we use collect to remember collenchyma. It means in collenchyma we'll be talking about collection of herbs, that means herbaceous plants, like talking about these vegetables like the taleno. So okay, how will you remember collenchyma? The first thing is that the letter CO, use it to remember cortex. So it means you see cholenchyma in the cortex. You know, when you draw the either the roots, the transverse portion of the root. So the center here is the cortex. So that's the cortex. Okay, so we said cholenchyma is found in the cortex of especially stem and leaves. We see it in the cortex of stems and leaves. Then another thing is that it is for herbaceous plants. You see cholenchyma majorly for herbaceous plants. So like I said, use collect to remember, as if you want to cut vegetables, you say let me quickly collect these vegetables. So it means for herbaceous plants. Another thing is deposit of cellulose. Cholenchyma cells, they have deposit of cellulose still from that letter C. So C-O-L-L, turn it to C-E-L-L, cellulose. So meaning that it has deposit of cellulose in their cell wall. Then they are living. How will you remember living? The L in the colon to remember living. Or you say any environment where you see that they are collecting herbaceous plants, means people are living there. So that's why they are collecting herbaceous plants that they will eat. So it means cholenchyma, they are living cells. Then they are elongated. Elongated, you can pick it from the E-L that is in this cholenchyma. Remember, elongated. So or you say because they eat herbaceous plants, vegetable, their lives will be long, so elongated. So then they are flexible. It means they can bend without breaking. That's why you see that all these vegetable plants, they bend when wind is blowing, but they won't break. They can bend. So that's flexible, meaning they can bend without breaking. So and how will you remember there? So you can use that same vegetable that yes, because we are collecting vegetables, so they don't always bend. So that is for or you just use the idea of when you want to collect something, you bend. You want to collect the herbs, you want to cut vegetable, you bend to cut it. So bending, so they can bend without breaking. So what are the functions of cholenchyma? One, for strength. So cholenchyma is for, you can turn the C of cholenchyma to S and say they are for strength. Or you say you eat the herbaceous plant vegetables so you can be strong. Then also for photosynthesis. Photosynthesis, why? Because they have chloroplast. You can use from the word cholenchyma to remember chloroplast. So for photosynthesis, or the green vegetable is for, it has for it undergoes photosynthesis. Then they have flexibility, talking about their bending ability, resilience. So that leads us to sclerenchyma. Once you remember skeleton, so you use it to remember sclerenchyma. So skeleton. So and take note, sclerenchyma is divided into fiber and sclerid. Just say selfish, SFS, S sclerenchyma, F fiber, S sclerid. So from this sclerenchyma, the first thing is that their tissues are dead cells. Dead cells, unlike the cholenchyma that we said they are living cells, their own are dead cells. How will you remember? Just say sclerenchyma from skeleton. Skeleton has no body. You just say this skeleton is dead. So dead cells. They are made up of dead cells. Then since I said they are divided into fibers and sclerid, what can we say about fibers? Fibers are elongated. You can remember from the letter R of this fiber, because fiber can be used for making rope, and rope is a long substance, so meaning that they are elongated. But sclerid, you can say they are roughly spherical. Take the S of the sclerid for spherical, R of the sclerid for roughly, so roughly spherical. Then another point is that they have lignin, deposit of lignin. Both of them, both the sclerid and the fiber, they have lignin. And anytime you hear lignin, that's something that is strong, that makes substance strong. So, meaning from sclerenchyma, you can remember letter S for strong. So they have lignin that makes them strong. So these are their general functions. Fibers, we use them for making ropes, like I said, letter R, ropes or twines or textiles, that's clothes. The fibers, they give flexibility. You can use letter F of the fiber to remember flexibility, or you remember rope can bend, so flexibility. Then sclerid, you can take letter S and C of the sclerid to remember seed coats, so they make up our seed coats. Then also letter S again, use to remember shell, so they make up the nut shells and the bark of trees. So that is sclerid. Then finally, sclerenchyma tissues occur in vascular tissues, corticals, and so on. Now that leads us to xylem, another supporting tissue. If you want to remember xylem, just say play xylophone on TV. Play xylo, the xylem is xylo, then phone, turn it to F instead of PH, on TV. So what do I mean by that? So looking at, there are 4 types of this xylem from the TVFP I wrote here. So that means from the play xylophone on TV, where the play will become parenchyma. It means we have xylem parenchyma, that's the play. Xylophone, the F, turn it to fiber. We have fiber in xylem. Then television, that's T, trachea. We have trachea in xylem. Then V, we use it to remember vessel. There's vessel in xylem. So looking at these 4 things, if you want to consider trachea, from trachea they are dead elongated cells. Dead, use the D of the trachea to remember dead elongated cells. Then tapering, use the T of the tapering. That means tapering means they are pointing upward, tiny. That means they were fat under, but by the time they get up they will be thin. So tapering. So end was, and again they are lignified. That means lignify means they are becoming solid. So like xylem, they are strengthened. Letter X, turn it to S, so for strengthening. So they are also lignified. So and they contain pits. So they contain pits, so that, or okay, another thing is this person I drew here. So let's assume this is the trachea, where air is passing. Trachea, windpipe. So trachea, I use it to remember trachea. So see the trachea as if it's a pillar. So pillar is for strength. So mean that trachea also is for strength. Then another thing is, we said there is pits. Pits, see that passage as a pit where air, water, oxygen, carbon, mineral salts, where they can pass. So mean that all this can pass through that same trachea. Then the next one is the vessel. So vessels, they help in upward transport of water and minerals. Upward transport, you know, when you write letter V, so it's pointing up, so you can say upward. Turn the V to W, so upward transport of water. Then bring it down, turn it to mineral. Upward transport of water and mineral, that's work of vessel. You know, vessel is also like container of water. So they also give strength and support to the plants. Strength, remember we have said X of xylem, turn it to S for strength. Then the function of fiber now is for tensile strength. Remember that fiber, we have rope. Rope is for tension, that means something that can stretch. So tensile strength. So summary of xylem is that it's for water and mineral salt. And take note, if you compare xylem to phloem, phloem sound like food, phloem, food, then it means xylem will be water. Or you can't read A to Z, WX, water for xylem. So it conducts water and mineral salts, taking it to the from the root. You take it to the stem, to the leaves, flowers and fruits. They can support the plant wherever it exists. Remember, it can form wood, so it supports the plant. Then the next thing we look at is phloem, which I just talked about now. If you want to remember phloem, just remember fly or flow. At rapture, Christians, they will fly. That means they will be caught up, both the living and the dead. That means those that are alive, they will be taken. Those that are dead, that are born again, they will be caught up. So it means phloem is a mixture of living and dead cells. And again, they are elongated. Use the LO of the phloem to remember long, elongated. Then tubular, you can say tube, flowing tube. Since I said use phloem to remember flow, something that will flow through the tube, so it means they are tubular. Then the different phloem we have, there are 4 types of phloem. So you can use escape. So escape sin, after after rapture, it means one will be escaped. So escape from sin. So we don't need the E, but we need the S, the C, the P and the S of the escape from sin. So the letter S means sieve tube. It means you will be sieved, you'll be checked before you go to heaven. Sieve tube. Then C, companion, you have people that will go with you. Then P, phloem parenchyma, so your parents, if they are born again, they will go with you. Then S, from that word sin, so that's sclerenchyma. So these are the types of phloem. So then don't forget, sclerenchyma is always fiber and sclerate. That's from cellfish, SFS, sclerenchyma fiber and sclerate cell. So both the phloem fibers and sclerates, they have secondary cells with that is thickened with lignin. So take note that this one also has lignin, since sclerenchyma from letter S to remember strong, so they also have lignin. So the major function is that the sieve tubes will help to transport organic solid food. So sieve tube, you know, when you sieve something, is that so that there can be a passage of free flow, just like in the drainage. So this one is sieving food. So from the sound, phloem is for food. Then the companion cells, they will associate, they will support that sieve tubes. Then phloem also can do the work of mechanical strength. So remember that phloem, I said the sclerenchyma in it can be for strength. Okay, so the next one, we now look at mechanism of support in plants. What are the things that support themselves in plants? So that support, you remember skin. Use skin to remember support. So, and that's why the first thing we talk about is the epidermal cell, from the word epidermis. You know, the outer layer of skin is called epidermis. So the same way plant has its own epidermal cell, its outer layer found in leaves, outer layer of stem, of roots, piliferous layers in root. Just say sprout, S for stem, P for piliferous, R for root. So sprout, stem, piliferous layers in roots, and then leaves. So that's where we can find epidermal cells. So it prevents the inner cells from injury. Remember that they are outside, they are supporting, so they don't want injury for the stem, the leaves, and the like. The infection, they don't want that leaf to be infected, and water loss, they don't want water to be lost from the leaves, and so on. Then there is no chloroplast in these epidermal cells. And how will you remember that there is no chloroplast? If you go and check the structure of leaf, which I'm still going to talk about when I get there. So there is a mnemonics I use there. So, and that mnemonics is from EMV. That means every movement is excellence. Every movement is excellent. So the E means what? That's the epidermis. We have the upper and the lower epidermis, while the M is the mesophyll. We have the palisade mesophyll. Then if you come to this place, there's another mesophyll, but we call that one spongy mesophyll, while this one is vascular bundle. So from this epidermal, so once I get here, I may not talk about this again. So the first one, which is letter E, which I call the epidermis, the upper epidermis, that's the outer layer of the leaf. So this is structure of the leaf. So outer layer of this leaf, so it's covered by a cuticle, something like a nylon that will prevent water loss. So that's epidermis. So there is no chloroplast there for photosynthesis. But if you come down to the palisade chloroplast or the palisade mesophyll, like I said, EMV. So the M is the mesophyll. So once we come to the palisade mesophyll, that one has a chloroplast. You can use letter P to remember the photosynthesis. It means for photosynthesis, that's for the palisade mesophyll. Then if you come down after the M, so you come to vascular bundle. So once you have vascular bundle, that's the xylem, the phloem, the cambium, and the likes. So that's in the vascular bundle. Then when you come down again, you see another mesophyll, which we call spongy mesophyll. This one also has chloroplast, but its own is not as much as palisade, because you from letter P, photosynthesis, that one we have enough. But this one is spongy, so it doesn't have enough chloroplast, though it's there. Now the last one that here is the lower epidermis. That one too doesn't have chloroplast, but especially those ones that are having stomata. You know, this place is surrounded with stomata, and then there is a guard cell at the entrance. That guard cell has chloroplast. So that's the only part that has chloroplast in the lower epidermis. So and that's why, back to what we were saying here about the epidermis. So we're meant to understand that from this epidermis, okay, so it does not contain chloroplast. So I've said it that the epidermal cell, they don't have chloroplast except in the guard cell, which I've explained just of herbaceous stems like the water leaf. So the part between the epidermis and the vascular bundle is the cortex. So when you say epi, that's outside. So the cortex is the one that separates the epidermis from the vascular bundle. Just like when you draw this, so it means you first encounter epidermis outside here, the vascular bundles, they will be in the middle. So cortex is now in between. So cortex is in between. So you can say ECV. So that's it. Then the parenchyma consists of large thin walled cells with many air spaces which permit air. So from the stomata. So for parenchyma, it can allow air to pass, just like our parents can be fanning their children, so they permit air from the stomata. Remember, stomata is for respiration, or lenticels to circulate to all parts. Now in dicots, dicotyledonous stems like sunflower, cambium is present between the phloem and the xylem. How will you remember? Just say XCP, as if you are spelling escape. So it means The cambium is between the xylem and the phloem. So cambium cells, they are constantly dividing cells and they help increase the number of cells, and that process is called secondary thickening. So what are they saying is that if you have 3 layers, where let's assume this is the xylem, the first layer, the second layer if it's the cambium, then the third layer will now be phloem. So mean that the cambium that is in the middle, it always expand, and as it's expanding, it will make the tree very large, and that's why we call it secondary thickening. So it's responsible for the increase in size of the trunk of many trees. Then we also have the pitch. So the pitch is the central part of the, it's just like when something enter the pit, so central part of the stem, and that's why you see when you draw a stem like this, so you have something at the center, so that's the pitch, so central part of the stem, and it's composed of parenchyma. So majorly parenchyma, you can say parenchyma like our parents, you can rest on their pits, that means the center part, that means you rest on them, trusting them. So then another thing is, if it is herbaceous plant, those small vegetable, so their own strength and rigidity is made possible by two-god pressure and supporting tissue. That why will vegetables stand firm? So it's because there is a two-god pressure, kind of water entering them, making them to stand, and there are supporting tissues like cholenchyma, parenchyma standing, helping them to stand. So that's through two-god pressure, and that's why there's something we call hydrostatic support, so it will support them by water. Then another thing is wood from xylem. So wood will make your stem strong and rigid. That's why you see some trees, they can, or generally trees, they stand because they have enough wood in them that will carry the branches, the leaves, and prevent them from being broken into pieces by strong winds. So by strong winds, so when strong wind is blowing, so it will not allow them to fall. So hence more wood a stem has, the more wood a stem has, the more difficult it is to break it up with the hand. So generally the function of all these supporting tissues we have been mentioning is that they give shape. So it means when you see a tree standing firm, it's as a result of the supporting tissues or this cholenchyma that gave it shape. Then also rigidity, they are firm because they have support from sclerenchyma, cholenchyma. They give them rigidity and strength. Then they make the stem of herbaceous plants resilient, flexible. You see that wind is blowing and all these herbaceous plants, they are not breaking, they are just bending. It's because there is supporting tissue there. Then they protect the delicate parts. So all those cambium, they will protect all those delicate parts. Now when we go into the internal structure of herbaceous stem, like all those vegetables, so what are the things you see, especially from the dicot stem? So from the dicot stem, once you get dicots, you can talk of all these trees like mango trees. So if you go into it from the structure, something like this is the structure of the cross section. Now from the first thing you will see is epidermis, that's the outer part. Just use the idea of assuming this is a tree, you know there is a bark. So that bark, compare it to orange. When you peel orange, you know the outer one, they call it epicap. Epicap is what you peel from orange. So instead of epicap, this one, the bark of the tree, so you call it epidermis. So as if you are just peeling, you do not cut everything, just peel. So epi to peel, so epidermis. Then when you now cut deeply, maybe for herb, so that cutting deeply, use it to remember cortex. So cortex, so cutting deeply, it means after epidermis is cortex, then pericycle. That means after cutting out the bark for herb, you will now see a clean flat surface, a clean surface there, which I call pretty pericycle. So it means that clean surface goes round, so pericycle. So pericycle is the next thing after epidermis, cortex, then pericycle. So that means that pretty surrounding part, pericycle. Then you move to vascular bundle. That means after cutting that pretty surrounding part, you will now come to the major center of the tree where we have vascular bundle. That means enough bundles at the center. Talking about the xylem, the phloem, then at the center you now see the pit and the medullary rings. Just the MP. Okay, so that's just how it is arranged. Now look at stomata and lenticels. They are found between epidermal cells. You know, epidermal is the outer layer of that plant. So and since you can see it in the leaf, and leaf can respire through stomata and their root through lentil cell. So that's why we find it there. Then the cortex is made up of cholenchyma. You can use cortex, cholenchyma. So they made up of cholenchymas and also parenchyma, or you say the cortex from cortex. Then cholenchyma, parenchyma, you can say copper. That means the copper is a cortex. So cortex has cholenchyma and parenchyma. Then the cholenchymas they form a strengthening hollow cylinder down the length of the stem, and the innermost layer of the cortex is bound by the endodermis. So once you hear innermost, that's endo is inside, so full of starch. Then pericycle, that one is composed of sclerenchyma. You can say PS like PlayStation. So pericycle is composed of sclerenchyma. It forms solid strengthening strands that run down the length of the stem. Then vascular bundles, these ones are arranged in a ring. So as you can see all this now, so vascular bundles, they are here. All these ones, they are surrounded. So it means they are in form of ring. So within the pericycle, so each bundle is made of phloem, cambium and xylem. That's vascular bond. Like I said, escape X, C and P. So they make up the vascular bond. There's also a pit. I said the center is the pit. So we have pit consists of thin walled parenchyma. It extends between vascular bundles as medullaries. So then that is also transverse section of monocot. We want to differentiate between monocot and dicot. For a monocot, like all these maize stand, so monocot stems, they show epidermis and the ground tissue and vascular bundles, they are scattered. So their vascular bundles they are scattered. As you can see them here, scattered vascular bundles. But this one is on, they are arranged vascular bundles. So the vascular bundles are surrounded by sclerenchyma. That's for the monocots. So it's sclerenchyma that surrounds their own vascular bundles. It's like the sclerenchyma is the savior of their own vascular bundles. Sclerenchyma saving the vascular bundle of the monocots. So then another thing is after that, the monocots, so surrounded by sclerenchyma, so which forms hollow strengthening cylinders that run down the length of the stem. So then cortex, pit and cambium, they are absent. So you can't see cambium in the monocot stem. So no cambium. That means cambium is always to make something very strong, very hard, and that's why your maize stand is not that strong. You can easily push it down. But for a tree, it has cambium, so it can easily stand. So that's for that one. Then another thing is the cholenchyma, sclerenchyma and xylem. They are the main supporting tissue in stem. You know why? Because the C, you can turn it to S for strength. S of sclerenchyma also strength. X turn it to S also strength. So it means cholenchyma, sclerenchyma, and xylem, they are majorly for strength. Then we have turgid parenchyma. This is an important supporting tissue in herbaceous stems. So for herbaceous, that means those plants, those ones that are like vegetables, so that are not that strong. So that means they have to be turgid. They take water to become strong. Then there's a woody stems like secondary xylem or wood. So that's the main supporting tissue. So we have ground tissues including all plant tissues except the dermal and vascular tissues. So the ground tissues include all plant tissues except dermal and vascular tissues. So thus the cortex, pit and medullary rays are part of the ground tissue. So in monocot we say cambium is absent. That means that thing that makes it strong is not really there. So in the vascular bond. So you can quickly look at these two pictures. One is for dicot, which is the one on the left. The other is for monocot, which is on the right. So if you want to remember dicot, the drawing, if you are told to draw a dicot stem, so you can say economist or echo is an economist, echo. So PEP, to remember people, the economist people, so they are crossing express. SP is express, so the economist people crossing express. The E represent epidermis, and that's why the first thing you see here is epidermis. The C is cholenchyma. The second thing you see here is cholenchyma. Then the P is parenchyma. The next layer is parenchyma. Then the E is endo, because we have entered inside, so endodermis. Then the another after the endodermis, then we also have, you can see escape from here, XCP, escape. That means the X is xylem, C is cambium, P is phloem. So meaning that the next thing you see is phloem. So from this label here, so you can see phloem. So the down part here is phloem, the middle will be cambium, then so this is the cambium going around, then the one that is on top of it here is now the xylem. Then pith is now at the center. So that's how to get the dicot. Then for monocot, when you come down to the right hand side, monocot, you can say escape, es for escape, then GVP, so escape through the ground. That means you dig the ground so that you can escape VP. Maybe there is a vice principal, so you want to escape through the ground, so escape ground VP. So E is representing epidermis, the outer layer epidermis. The S is the sclerenchyma. After epidermis, you see the sclerenchyma. So you know it's always surround the vascular bundle. Then the G, use it to remember the ground tissues. So this is the ground tissue, all those bundles. So after the ground tissues, then V, the real vascular bundle, the xylem, the phloem. So that's vascular bundles. Then P is the pith, which is the center. So that's the structure of the mono and the dicot. So if you want to differentiate between monocot and dicot, then just say TCP. You know there is one liquid they call TCP. So you can say TCP is very clean, where your T represents secondary thickening. So it means there is thick secondary thickening in dicot. You know dicot has the cambium, so it has cambium, as you can see here. Cambium is here, so which it will be using for thickening, expansion, but here there is no cambium, so that's secondary thickening. So if there is cambium, then there will be secondary thickening. So this one has cambium and the secondary thickening, but this one no cambium, no secondary thickening. So that's TC. Then P is pith. This one has pith, this one does not have pith. If you come up here, you can see that the pith is present here. So pith is present here. So though this one here, the root pith, so what we can just say that the pith is not that visible. So let's put it in that way. So the pith is not that visible, not that it's totally absent. So that's for TCP. Then VC, the V, you can use to remember vascular bundles. They are few and arranged in ring. So few and arranged in ring. So see the vascular bundles here, 1 2 3 4. So they are not much, they are few and they are arranged in ring round. But this one is own vascular bundles, see them scattered. So they are not in ring, they are just scattered there. So it means that the vascular bundles are in ring for this, but this one they are numerous and they are scattered. So then the last one, C, cortex. The cortex is wide, but this one the cortex is narrow. See the white surface, plain white surface as cortex, so it's wide enough. But this one there's not enough white surface because all these vascular bulbs are covered there. So that's how to remember. Though you still have other things that you can add to them apart from TCP. Very clear. You can say cholenchyma are below epidermis. So when we say cholenchyma are below the epidermis, look at the cholenchyma here is under here, epidermis is above, so cholenchyma is below. But for this other one, so for the other one, we say sclerenchyma are below epidermis. As you can see, sclerenchyma, they are below epidermis. So if you come here, you will see sclerenchyma is here. The epidermis is outside. Epi is always outside, so scleren will now be inside. So that's for another thing you can take note of. Then pericycle is made of sclerenchyma. That's for, if you remember, PS, PlayStation, pericycle is made up of sclerenchyma. But this one, sclerenchyma is surrounded by vascular, like I said, savior. Sclerenchyma is surrounded by vascular bundle. So that's for that one. Now, from the next one is roots. Anytime you see a drawing that has something like this, that's roots, as if there is air is roots. So this one is for dicot, this one is for monocot. I told you if you want to remember dicot, like a tree is a dicot, maybe mango tree. Why monocot is like your maize stand. So maize stand. So let's look at the structure of each of them. Now from this place, you can see this. Okay, just let's start from this one. Price. Just say maybe you want to buy Windows SP. And you try to get the price of that SP. So the P means piliferous layer. So this round is the piliferous layer. Then R, that's the root air. You know, air always stands. So root air, piliferous layer. Then the C will be the cortex. You know, that surrounding here is the cortex. So this space is the cortex. Then X, xylem. So this one that is a xylem. Then phloem, these tiny ones. So that's for the monocot roots. So monocot roots. So let's now check it. So the internal structure of herbaceous dicot roots. So for dicot, what are the things we can see similar to that of monocot? So they also have piliferous layer. So in its own piliferous layer, there is no cortical there. So it's no cortical that will aid water absorption. So no cortical, and it will aid in water absorption. That means it will not be conserving water. So that's piliferous layer. So this it also has its own piliferous layer that does not have cortical. That means something that looks like nylon is the piliferous layer, I mean the cortical. Just like leaf has cortical, so this one it doesn't have cortical. Then apart from piliferous layer, we also have cortex. Cortex is made up of parenchyma. So cortex is made up of parenchyma. So parenchymatous cells and helps to store food and water. Remember, our parents, they get food and water for us. So it means you can say cortex has parenchyma. So that means maybe you can say copper, cortex, parenchyma. So or PC, personal computer. So just find a method of remembering. The endo, inside endodermis. So this one is between pericycle and cortex. So you know endo is inside, peri it will surround, so between pericycle and cortex. So that's where we find your endodermis. And endo, it is starch that is inside. Pericycle, so that one that's where root is coming from. So you know if root will come out, so the way the roots will come from is called that's the pericycle. Then vascular bundles, we have xylem and phloem for vascular bundles. So it means that's where water and food will be passing. The internal structure of herbaceous monocot root is similar to that of dicot roots with few differences. So let's quickly look at the differences between internal structure of a dicot root and internal structure of a monocot root. So for a dicot root, pith is absent. When you say dicot root, this is it here. So there is no pith at the center. So pith is absent. So for pith is absent in the internal structure of a dicot root, but for monocot root, pith is large and well developed. So pith is large and well developed. So as you can see, there is enough space. Or just use this idea that when we're doing stem, we said for the dicot, so it has pith. So pith was large enough in the dicot, so but it was small in monocot. But reverse is the case when it comes to roots. So pith will now be absent in the dicot and it will be large in the monocot. So that's number one. Then number two, secondary growth occurs leading to development of cambium and cork cambium. So it means the same thing that happens in stem will still happen in the root of this cambium now. So it means the cambium here is still strong, is still large. So that means that's where there's development. But for monocot, no cambium. We said for mango tree, we will see cambium that make it strong, but this one no cambium. Now few phloem bundles are present in dicots. So few phloem bundles, but in monocot many phloem bundles. As you can see here, phloem, this is the phloem, there are few, 1 2 3 4, but this one phloem, there are many phloem bundles are there. So then it has only xylem bundle which is star shaped. That's for the dicot. So they have star shaped xylem, and centrally placed. That means is at the center. So the xylem is at the center and it's like star shaped. It's like star shaped and it's at the center. That's number 4. So for this xylem, so meaning that from this drawing here, you can see the drawing here that there is a star shaped xylem here. So there is a star shaped xylem, so at the center on the left. So this is it here, you can see it moving around there. So while on the right hand side, so there is no the xylem there is not star shaped, but on the left it is star shaped xylem. So meaning that for this we say it has only it has star shaped xylem and it's at the center, but on the right, the monocot, his own xylem bundles, there are many. So many are radially arranged. So here now you can see the xylem, there are many and they are radially arranged. That means around, unlike the xylem of this that is just star shaped. So that's for the xylem. Then there's also what we call xylem vessels. They are angular, but this one xylem vessels, they are oval. So that means this ones, they are like angles, like this is an angle, this is an angle, but this one is oval, round, so something like round. So that's for the xylem. So then the last thing for today is the leaf. So we want to consider the leaf, and that will be the end of this class. Now, internal structure of a leaf. The internal structure of a leaf consists of three main tissues. There are three tissues in our leaves. So I've actually explained this earlier, but I'll just summarize it that you can say every movement is excellent. The E, that's the epidermis. I said we have upper and lower. The upper has cortical that preserve water or conserve water, and there is no chloroplast there, no photosynthesis. So then M, that's mesophyll. Mesophyll is divided into two. Palisade, letter P, it has photosynthesis or chloroplast. Then the other mesophyll, that is spongy mesophyll, that one also has for chloroplast for photosynthesis, but it is not much. It is not much. So then after EM, then we have V, vascular bundles. So it means there are also vascular bundles like xylem and phloem in our leaf. So the leaf is covered with a cortical. I've said that, with preventing loss of water. So the outermost layer is the epidermis. We have mentioned that lower epidermis, there are enough stomata. So if you come to the lower epidermis here, stomata are there where there is gaseous exchange, and there is a guard cell there that always change the shape of this structure. So, and looking at the leaf, so if you look at structure of a leaf, it has a stalk. So that's where there will be a passage of the water so into the mid rib, and then scatter through the vein, all the network veins of the leaf. So that's how water moves around the leaves. So this is where we stop for today. Thanks for listening.